Hello everyone, this is Sapna from RPIIT Medical and Technical Campus. In today's class, we are going to discuss about colorimeter. So what is colorimeter? In colorimeter, color Colori indicates to color and meter indicates to measure. It means colorimeter is an instrument which is used to measure the color intensity of any reagent. And on the screen we can see the image of a colorimeter. So now we will discuss about colorimetry. The process of measuring color is known as colorimetry. Means it is the most common analytical technique used in biochemical estimations in clinical laboratory. It involves the quantitative estimation of color. A substance to be estimated colorimetrically must be colored or it should be capable of forming a chromosomes. Chromosome means colored complex through the addition of reagents. Now we will discuss about the range with or the rays which can a colorimeter measure. A colorimeter can only measure the visible rays which are in between 380 to 750. Here we can see the wavelength in nanometer and reason and observations. Less than 380 nanometer wavelength come into the region of ultraviolet, and these are invisible rays and our colorimeter can't measure them. In between 382 to 440, these are visible rays and come under the violet filter, and our colorimeter can measure them. From 440 to 500 nanometer, these are also visible rays and comes under blue filter. 500 to 580, these are visible rays and comes under green filter. 580 to 600, these are visible rays and comes under yellow filter. 600 to 620, these are also visible range and come under orange color. 620 to 750, these are also the visible range and observed under red color. But more than 750, 800 to 2500, these are non-infrared rays and these come under the not visible. Means these are also not visible in colorimeter. Now we can see the diagram of colorimeter. It has a light source, then a slit, then a lens and a filter, then a cuvette and photon cell and a output. We will later discuss about its part and their functions. Now we will discuss about the principle of colorimeter. The principle of colorimeter is based on two laws. There are two scientists. These are Barrier and Lamberts. They given their different laws, and by combining these laws we makes the principle of colorimeter. So according to Barrier's law, when a monochromatic light passes through a colored solution, amount of light transmitted decreases exponentially with increase in concentration of a colored substance. It means that is the amount of light absorbed by a colored solution is directly proportional to the concentration of substance in a colored solution. And next we will discuss about the Lambert's law. Lambert's law says the amount of light transmitted decreased exponentially with increase in path length, means diameter of the cuvette, or thickness of colored solution through which light passes. That is means it means the amount of light absorbed by a colored solution depend on the path length of cuvette, that is 1 mm in thickness in diameter or depth of a colored solution. These both law combines and makes the principle of colorimeter. Now we will discuss about the parts of colorimeter. First of all, we can see light. Light is made up of tungsten filament lamp is used in a colorimeter which emits a continuous spectrum of light in the visible range of 400 to 760 nanometer. Then we can see the slit. It is a adjustable which allows only a beam of light to pass through. It prevents any unwanted or stray light. Then we can see the condensing lens, and this condensing lens, light after passing through the slit falls on condenser lens, which gives parallel beam of light. Then we will talk about filter. Filter is made up of colored glass or dye, gelatin filter sphere used for selecting the light of narrow wavelength, that is 50 nanometer or more. Filter will be absorbed light of unwanted wavelength and allow only a monochromatic light to pass. For example, green filter absorb all colors except green light, which is allowed to pass through the light passes through a green filter have wavelength of 500 to 560 nanometer. Filter is used always complementary in the color to the color of the solution. Then we can see the cuvette holder. It means sample holder. The monochromatic light from the filter passes through the colored solution placed in cuvette. It is made up of a special glass, plastic, silica, quartz material, etc. It may be square, rectangle, round shape with a fixed diameter of 1 centimeter, have unformed surface. The colored solution in a cuvette absorb a part of light and remaining is allowed to fall on detector. For example, a solution of red color transmit red light and absorb complementary green color. The width of cuvette represents the path of light. Now we will discuss about the photocell. Photocell, its another name is detector. Detector are photosensitive elements which convert light energy into electrical energy. The electrical signals generated directly proportional to the intensity of light falling on the detector, usually photomultiplier tubes, which are based on photoionic effect used as a detector. Now we will discuss about the readout device. The electrical signals are generated in photocell is measured by galvanometer, which displays the percentage transmission and optical density. Thank you.